Good evening, everyone. This is, as promised, the part 2 of the chapter Life Processes for Grade 10 CBSE, and it is based upon NCERT information. So, we are going to split nutrition into two plants. Today, we are going to deal with how plants and other organisms, simpler organisms, obtain food and generate their own energy. And the third part of the series will be about nutrition in highly organized animals such as we humans. So, well, we already are familiar with the term nutrition. Nutrition is apparently the kind of life process which helps us to obtain food for energy and for synthesis of protein and other substances, either from the surrounding environment or generation of this carbon-rich source. And of course, this entire process of obtaining carbon-rich sources or food which will create energy, this process is called nutrition. And of course, depending upon different organisms in different environments, the modes of nutrition are also variable. So the first case is that there are certain organisms which can generate or synthesize their own food resources, their own carbon-rich compounds. So they are the self-synthesizing organisms. They are called the autotrophs. Again, split the term. Auto means self. Trophy is that this is their means of nutrition. Right? So they are autotrophs, and this mode of nutrition where the organisms are synthesizing their own carbon-rich organic sources of food, this is called the autotrophic mode of nutrition. And of course, they are able to synthesize the organic food from inorganic substances like ions and atoms, etc. So under autotrophic mode of nutrition, we have two main categories. The first category is photoautotroph and the chemoautotroph. So photo means light, photon from the word photon, light. So these are organisms which are dependent upon generating their own food resource in the presence of light, right? So photosynthesis, which most of the plants or the pigment containing organisms undergo, is a phenomena under photoautotrophism. And chemoautotrophs are those organisms which don't require light. So they are having means of utilizing organic compounds, of making organic compounds from oxidation of chemicals or inorganic substances. So they can directly synthesize their own chemicals, but they don't require light, right? So they derive their energy from various chemical reactions, right? They can include cyanobacteria or the blue-green algae, sulfur oxidizing bacteria. There are certain extremophiles, certain species of bacteria which live in very extreme habitats of high temperatures, low temperatures, very acidic or basic environments, and they are the ones which can generate their own chemical food resources. Like certain group of bacteria can generate their energy from sulfur, from nitrogen, from iron. So I hope this is clear to you. So autotrophic mode of nutrition will include photosynthesis and chemosynthesis, two main processes. And then we have the consumers, which are the heterotrophs, which practice the heterotrophic. So hetero means others. So they are dependent upon other organisms for getting their carbon-rich energy source or procuring their food. They cannot synthesize their own food. And well, all the consumers, they are under this category. And the consumers are there that they will receive organic substances, whether from the living or recently killed organism. So if there is an organism which eats or feeds on greens or on vegetation and plants, that is a herbivore. If they are dependent upon preying and killing other animals, like let's say a lion or a tiger hunts, right? So that is a carnivore. And they are then the scavengers like the vultures. right? So these can have different categories under the consumers. You can have a number of categories. Detrivores, another category, which will ingest non-living organic matter or dead organic matter, you can say. Or, for example, earthworms in the soil. They are a source of feeding on detritus. So the word detrivores comes from the word detritus. Detritus means dead, right? So earthworms in the soil are a good example of that. And then the saprotrophs. So sapros means non-living organic matter, decaying organic matter. And they have a way of ingesting the dead organic matter. How? They first secrete the enzymes, the digestive enzymes. Remember, enzymes are the biological catalyst. They help in the breakdown of big molecules, complex molecules into simpler forms. So they are secreting these digestive enzymes, and then once they are broken down to simpler products, they just absorb the simple products. Now, a lot of fungi, a lot of bacteria are under this class of decomposers and saprotrophs. And when we talk of these fungi and when we talk of these saprotrophs, because they are releasing the enzymes and out of their cells onto the dead decaying matter to break it down into simpler substances, this becomes a form of extracellular digestion. So your assignment is differentiate between intracellular and extracellular digestion. So your hint is that in the previous video, I have shown you via a video a beautiful example of intracellular digestion. That is digestion taking place within a cell. Digestion usually means the breakdown of complex molecules into simpler molecules to create energy. And yeah, for extracellular, you have a big example right in front of you. So coming back to the NCERT text. So in this paragraph over here, how living organisms are obtaining their food, this has been generalized by me in the form of a flow chart. And I've also told you about the autotrophic mode of nutrition, that you can have the photosynthesis and chemosynthesis, two kinds of processes, and the organisms can either depend on light or they can simply, convert the chemicals and, can do their chemical synthesis of inorganic substances and utilize them directly in their metabolism. So all of this can be studied over here. So what are the crucial parts over here? For example, when we say photosynthesis, so photo, I'm again repeating, photo comes from take the word in biology, photo from photon, that is light. Synthesis means production of something, right? So this is the process in which the autotrophs will take up inorganic substances from the atmosphere, surroundings, and then they will convert them into stored forms of energy. Now, usually, what are they utilizing? They are utilizing, number one, carbon dioxide. Remember, we are generating a lot of carbon dioxide because of respiration. So whatever carbon dioxide we are generating, the consumers are generating, the autotrophs are utilizing that in photosynthesis. So that's the circle of life. And then, of course, there is water. I'll tell you about the breakdown of water and its relevance. And this entire process happens in the presence of sunlight in the photosynthetic active radiation region, or PAR. So go ahead and find out. That is the second assignment question today. What is PAR? P-A-R. And what is its significance? So they are then converting them into carbohydrates in the presence of sunlight and chlorophyll. Well, see, in the presence of chlorophyll, light is getting trapped in the cells because of these pigments called the chlorophylls, right? And these pigments are present in cell organelles called the chloroplasts. Do you recall where they are present in the chloroplasts? In the stat coin-like structures called grana, singular granum, which are made up of membranous structures called the thylakoids, flattened disc-like structures, and it is in the thylakoids of the grana that the pigments or the chlorophylls are present. So the third assignment question is, draw the diagram of the chloroplast. This is a previous knowledge skill, so I'm not repeating it again. You have done it in class 9. Draw the diagram of the chloroplast, label the parts containing the pigments, and let's see what you draw for us. So this carbohydrate, the stored energy, which will be created as a result of fixing the carbon dioxide in the presence of sunlight. This carbohydrate is starch in case of plants. And in our bodies, we also have fixed sources of energy and carbon and carbohydrates. So in our body, the fact that we are not performing photosynthesis because we lack the pigments and we lack the enzyme machinery for the same, and we can't trap sunlight. So we are heterotrophs. But even in our body, the reserve carbohydrate is glycogen. So you have a very good question over here, that what is the reserve food material in plants and animals known as? Both of them are different forms of carbohydrates. I'm sure you are going to go through them. Okay, which brings us to this most classic equation of photosynthesis right over here. So essentially, this figure, this chemical equation mentioned in your textbook about the generation of glucose, which is the basic sugar formed as a result of photosynthesis, this is actually the overall crux or the summarization of the entire complex chemical reaction called photosynthesis. So if I were to tell you, let's split the entire process into two kinds of reactions, namely the dark reactions and the light reactions. What does that appear like? So I'm going to make you understand that there's a leaf, right? In the leaf, we have different kinds of cells. In the cells, we have chloroplasts, right? So in the chloroplasts, we have, if you recall this structure from your class 9, the chloroplast has a matrix, right? And it has the stacked structure called the grana. The grana is made up of the flattened discs called the thylakoids. And in the thylakoids, you have the pigment called chlorophyll, right? And of course, the stroma is the matrix again. Right? So the matrix is the place where the dark reactions are going to take place, and the grana is the place in the entire cell organelle called chloroplast where the light reactions will take place. So what are the light reactions and what are the dark reactions? For that, we come back to our text. In our text, photosynthesis has been summarized in the form of 3 steps, 1, 2, and 3, right? So the first step is that in the chloroplast, in the grana, light in the visible radiation region will be trapped by the chlorophylls, right? This trapped light is going to cause the splitting of the water molecules to release oxygen and hydrogen ions. So your assignment is, give the chemical equation for the splitting of a water molecule in the presence of light. In other words, I'm asking you to give me the chemical equation for photolysis of water, the breakdown of water molecules in the presence of light. And this releases oxygen and hydrogen. Now this reaction is very crucial. This breakdown of the water molecule in the presence of light. Step 2. This is a part of the light reactions now. The first kind of reactions, right? Of photosynthesis. This is very crucial because this will help in the creation of certain energy-rich phosphate compounds which are going to be utilized in the third step over here. So that is your linking. At the level of grade 10, it is beyond the scope that I discuss with you the details of the light reactions, that how this oxygen will be released and how the hydrogen ions are going to help, and then the fixing of the phosphate-rich compounds and how those phosphate-rich compounds like ATP, etc. are going to be utilized in the third step. But this is how I can give you a connection. That the light reactions will occur in the presence of light, So essentially the first step is the splitting of the water molecule to release oxygen and hydrogen ions, and the creation of some rich, phosphate-rich compounds which are going to energetically drive the third step. So the carbon dioxide from the atmosphere is going to be reduced within the plant cells, within the chloroplast stroma matrix. Remember, just in the video you saw right now, matrix of the chloroplast, the stroma. Over there, the carbon dioxide will be fixed or reduced in the form of carbohydrates. What kind of carbohydrates? Glucose. A lot of glucose molecules will be generated. And at one time, so many glucose molecules may not be utilized by the plant. So they will be polymerized in the form of a reserved food material or the reserved complex carbohydrate or sugar called starch. I'm sure all of you have eaten starchy foods like potatoes, right? So whenever you're cutting potatoes in water, you see the water goes milky white. That is the presence of starch, right? So here's figure 6.1, which gives us a very good description of how are we talking in terms of the anatomy or the inner tissue organization of a leaf. So from the lower classes, you know that the leaves have been called food factories of the plants. Where are the leaves located? On the stem, on the shoot. And we know from junior classes that they're the food factories. Why? Because the leaves are green. The leaves contain cells. These cells contain a lot of chloroplasts and chloroplasts, a lot of chlorophyll. So now you know light reactions are taking place. But I forgot to tell you, what are the dark reactions? This step, this reduction of carbon dioxide to form carbohydrates, this happens by a series of complex steps which we call as the dark reactions. Now, what is the distinction? A dark reaction in photosynthesis necessarily does not mean that it will happen only in darkness. No, it's just a means of saying that it is not dependent upon the presence of light energy. Right? So it may happen despite the presence or absence of light. So if this is a leaf, and if you cut the leaf vertically and you get a cross section, what is the internal organization of different cells? What does the leaf look like internally? So the leaf has an upper protective layer called the epidermis. It is made up of rectangular cells tightly packed. In some cases, you may have a cuticle, another protective layer beyond the epidermis. So the upper leaf surface will have upper epidermis. The lower leaf surface will have the lower epidermis. And then underneath the upper epidermis, we have a layer of long columnar cells. Please, I apologize for the background noise. If you can hear that. Difficult times in quarantine, remember guys and girls. So this layer of cells, tightly packed columnar cells just beneath the epidermis, upper epidermis, has been drawn in your NCERT but left unlabeled. So I request you to label it as the mesophyll cells. M-E-S-O. Meso, E-H-Y-L-L. Mesophyll cells. Because these are the cells which are densely packed with the chlorophyll containing organelle, the chloroplasts. So now you know where the light energy is getting trapped for the light reactions. And they are a bit tightly packed, so you can eliminate these spaces in the diagram. This is followed by almost spherical or disc-like cells, loosely packed. And this is called the spongy parenchyma. S-P-O-N-G-Y, spongy parenchyma. Well, these also have some chloroplasts and they have huge air spaces or air cavities in between. And then, of course, we have the next layer, which is the lower epidermis. Now, if you have an observant eye, you will notice that the lower epidermis is different from the upper epidermis. The tiny pores called the stomata or the stomatal pores present, and the cells which guard these pores are called the guard cells. Students, I'm highlighting it right now. So I'm saying that the spellings are G-U-A-R-D, guard cells. please do not commit the error of writing GAURD. I know it sounds very strict, but in biology, a subject where the entire context is with reference to the terms and the meanings of those terms, you should not be getting the spellings incorrect. So I hope you will keep this in mind. So because they guard the pore, the opening, they're called the guard cells. And now you understand the reason why the spongy parenchyma has a lot of cleared spaces, because these are the pores which allow the movement of carbon dioxide into the leaf tissue. So the light is being trapped over here, carbon dioxide is moving in here. You have the perfect circumstances for photosynthesis to happen and for the plant to produce its own carbon-rich food resource. Now, this is a very important diagram, and this should be, as an assignment, practiced by all of you. So this is also to be drawn, and this is also to be practiced in your biology textbooks and notebooks. And remember, I have given you some labelings. And if you have a keen eye, you might want to consider that both these surfaces of the leaf are different from each other. So, I give you a previous knowledge assignment question over here. Again, too many assignment questions, huh? But what to do? These are times of quarantine and a lot of self-study is required. The question is, the angiosperms, the true seed-bearing plants and the fruit-bearing plants, the plants with the vessels, xylem and phloem. There are two types. Monocots and dicots, monocotyledons and dicotyledons. The monocotyledons means that the embryo has one cotyledon for reserve food materials, and the dicotyledons have two cotyledons in the embryo for the reserve food material, and they have a string of differences among each other. Let's say a dicot like garden pea or hibiscus, and compare that to a monocot like wheat, maize, rice, any grass, that's a monocot. So the question is, are the monocot leaves and dicot leaves different from each other with respect to their upper epidermis and lower epidermis? So this is the cross section of not just any leaf. There is a correction over here which I would like you to know. This is the cross section of a dicot leaf, also called the dorsiventral leaf. Okay, so go on, find out what is the difference between the leaves of a dicot and a monocot. The dicot has a dorsiventral leaf. What does the word dorsal and ventral mean? Hint is with respect to the surfaces, the upper epidermis and the lower epidermis. You see a clear difference, presence and absence of stomatal pores. Right? So I leave it to you. I'm sure you guys are very smart and you are going to work out all the challenges. So that brings us to the point. Right now, for now, we are skipping the activities. I will wait for the schools to open for that, and then we'll be doing them in the lab. So just wanting to show you under the microscope, how do stomata appear? So diagram is there in your NCRTs. And how does the stomatal pore open so that, air rich in carbon dioxide can move in for photosynthesis? And when does the stomatal pore close? Right? So this diagram is, from the examination point of view, a very, very important diagram. So you can very clearly see that the guard cells, when they are fuller, when water moves in from the surrounding cells into the guard cells, they become swollen up, they swell up, and the pore opens. But when the guard cells, they lose water to the surrounding cells, they shrivel or they shrink, and the pore closes. And how does that appear actually under the microscope? So I'm just going to play this small clip for you. You can see that this is a section of the leaf under the microscope. This is the surface, and do you see these two bean shape, these kidney-shaped guard cells, and this is the stomatal pore. Clearly, you can see that the guard cells are swollen up with water. These green dots, by the way, are the chloroplasts in the guard cells, and these big circles that you see over here are the nuclei of the guard cells. Compare this figure to that of the diagram drawn in the NCERT. You will find similarities. So the NCERT diagrams are standard. Do not skip them. So I'm just going to play the video and you will see how the guard cells are shrinking and the pore is tightening. See how the pore is tightening? And slowly and steadily, the pore is just a slit, right? So the pore is closed. At the moment, the pore is closed. So that brings us back to the NCERT text. In class 9th, it is already mentioned that you know that the stomata are the tiny pores which are present on the surfaces of the leaf. Now the tricky part is they are present on the upper surface or the lower surface, right? And if they are present on both sides, what kind of a leaf is that? If they are present on just the lower epidermis and the lower leaf surface, what kind of a leaf is that? In your NCERT, the crux is that the cross section of the dicot leaf or the dorsiventral leaf has been drawn. Okay, and massive amounts, large amounts of gaseous exchange of which gases? Well, carbon dioxide and oxygen. Oxygen will be released and carbon dioxide will be taken in, occurs for the synthesis of photosynthesis, right? And of course, gaseous exchange does not just happen across the leaf surfaces, but it also happens across the stem, along the roots, along the other plant parts as well. And because of this opening and closing of stomata, because of the stomatal pores being open to let the carbon dioxide move in for being reduced to carbohydrates because of photosynthesis, a lot of water can be lost. A lot of water from the surrounding sense can be lost to the atmosphere, and this phenomena is called transpiration. Water is lost in the form of gaseous vapors. This is transpiration. So transpiration has been termed as a necessary evil. And if the dorsiventral or the dicot leaves are having their stomata towards the lower epidermis, does this strategy help? So that is an assignment question again for all of you. And this diagram, again, I'm stressing out. It's very important for your examination purpose to draw the open stomatal pore and the closed stomatal pore. You can clearly see the distinction between the guard cells. Another important thing, if you have noticed, the lining of the guard cell around the pore is very thick, but the lining of the guard cell towards the other cells, the epidermal cells, is very thin. So there is a significance to it that water has to move in or water has to be lost from here. So the cell lining is very thin towards the epidermal cells. You have to draw the nuclei, you have to draw the chloroplasts. How is the stomatal pore going to open? Because the stomata are going to allow water to move in. The guard cells will allow the water to move in. They will become swollen up, they will swell up, and there is another term for it. We say the guard cells become turgid, P-U-R-G-I-D. You may note it down somewhere. And the pore opens. But when the pore has to close, to avoid excessive loss of water due to transpiration, the guard cells will lose water to the surrounding epidermal cells or the subsidiary cells actually, and they will shrink or they will become flaccid, F-L-A-C-C-I-D, and the pore will be reduced to a slit. It will close. So there are certain plants which open their stomatal pores at night and they keep them closed at the daytime to avoid this excessive loss of water due to transpiration. Can you tell me in the comments below which plants am I talking about, which will open their stomata at night and keep the stomata closed during daytime? So this is very important for you, this entire paragraph, this entire figure 6.3a and b. Oh, it's so important. And I would be closing the lecture with the electron images of how the guard cells appear So this is an actual image from under the microscope, and you can see the guard cells are bean-shaped or kidney-shaped, and this is your open stomatal pore, right? You can see the bean-shaped guard cells guarding the open stomatal pore, and over here the pore is like a slit, it's closed because the guard cells have shrunk, become flaccid, and the bean-shaped guard cells, the kidney-shaped guard cells, they are present in the dicot plant leaf, right? So the leaf surface of a dicot plant will show you this kind of guard cell, the bean-shaped or the kidney-shaped. If we are talking of the grasses, the monocots, now this is an advanced concept for those of you, who are going to prepare for NTSE. Look at this. Guard cells are not bean-shaped. Now this is the surface, the epidermal surface of a monocot leaf, let's say maize, rice, wheat. So the guard cells are dumbbell-shaped. They are elongated, just like the gym dumbbells, right? And pores are, of course, closed. Can see. So you have to tell me in the comments below, how are the stomatal pores and how are the stomata different with respect to their distribution on the upper and lower surface of a dicot leaf and a monocot leaf, and then how are the guard cells different in their shapes and their appearance under the microscope in the dicot and the monocot leaves. So I am sure this is it about the autotrophic nutrition, that is nutrition in plants. And so far we know that you require water for photosynthesis, right? But there are other substances too. So we know that the roots are in the ground and the roots are providing support and anchorage, but the roots of the plant also help it to absorb water and essential mineral ions from the macronutrients and micronutrients from the soil. So all of these micro and macronutrients, water, we know they are being utilized in photosynthesis. And of course, what are the macronutrients? Well, nitrogen, phosphorus, iron and magnesium, because they are required in very small amounts. They are the micronutrients. So you can just read through and serve through this area. And you do know that how the nitrogen fixation takes place, how certain plants have the ability to trap nitrogen from the air, which is N2, and convert it into nitrates and nitrites in the soil with the help of certain root-dwelling bacteria. And then, of course, these nitrates and nitrites are converted to ammonia, NH3, that form of nitrogen which is readily used by the plants to make amino acids and proteins and a lot of other molecules. So, well, that is beyond the scope of grade 10. For that, if you take medical class 11, we will be discussing that. And yeah, so that was autotrophic nutrition. So till now we know that you require plants, they require carbon dioxide, water, and light for photosynthesis. So these are absolute, and chlorophylls, of course, to trap light. So chlorophylls, light, water, and carbon dioxide. If you find time during this week, you can easily go through activities 6.1, activity 6.2, read them, and try and figure out how light, chlorophyll, and carbon dioxide are essential for photosynthesis. In other words, they become limiting factors, factors without which photosynthesis cannot occur. So I conclude the class for today. You have too much to learn and answer in the comments below and write in your biology notebooks. And we are going to pick up heterotrophic nutrition, especially with context to human beings and our digestive system and how that works in the next class. So all the best. Be focused, be self-motivated, and remain busy. Involve yourself in a lot of hobbies and some studies too during this quarantine. Happy studying.